Yeah, as I work for the genome browser, and this is a kind of special application of text mining, I might have to introduce the genome browser very, very quickly. So the UCSC genome browser started just after or slightly before the sequencing of the human genome. It's a pretty old website. It has not really changed since '99. It's heavily used by people that work somehow with the genome. So there's more than 20,000 citations and more than 100,000 monthly users. There's hundreds of tracks, and the whole project fits in very well into the topic of the workshop because the genome browser, being a kind of hub for genomic information, interacts with many databases. Not as much the ones here, but at least most of the human databases. Most databases you can find anything related to human proteins or genes is usually reachable or somehow in the tables of the genome browser, somehow mapped to the genome. Many people use it. It has not, so the basic usage pattern has not changed. The internal technology is still old school ANSI, C, and MySQL, and nothing has changed about it. That's why, I guess that's one of the reasons why it's relatively popular. So these users, they find something on the genome, and the idea is that once they have found exon number 2 of their favorite gene, they go to PubMed and want to know more about it. And that's why we would like to add more literature information into the genome browser. So usually they come from a genomic locus. They might have a gene of interest, but they still have their own topic of interest. And here comes in the triage aspect that MGI has. They have a topic. They're either clinical researchers, biologists, biochemists, very other flavors of biologists, and they have their topic of interest. And they use we all use document classification all the time, and we trust it. Databases might not like it a lot, but all these formulas and base and unigrams and bigrams, but they use it every day in the spam filters of their email messages. 90% of email on the internet is spam, and we use it all the time, and we kind of trust our spam filters now. So something similar should also work for research articles. That is very hard to do from abstracts, as you all know. It's a lot easier if you have the full text PDF, but text mining, you are not allowed to do text mining on full text papers, apart the PubMed Central ones that get mentioned here all the time. PubMed Central is 300,000 documents with PMIDs, around 600,000 documents in total. PubMed Central has 3 million PDFs on their servers, but are not allowed to let us text mine them, or they will not let us text mine them, or any one of us. The reason for that is the good old problem of copyright. Of course, authors sign away the copyright. We know the open access movement, and the terms of services of most publishers do not allow you to download documents in bulk. And that is getting better, but the basic problem persists that the publishers will not give away their property that's $1 billion industry for free. So what we did, what I did, I started to call all these publishers one by one, and I asked them if we could have permissions to download all their PDFs, and most of them said, fortunately, yes. So now we have around 4 to 5 million articles, with 2 million still coming in. That hasn't really changed a lot since the BioCreation meeting. And in total, that's around 80 to 100 gigs raw full text and a lot more of supplementary files. When I can get them, I will try to get the supplementary files as well as possible. These come in various document flavors, so PDF, Microsoft Office, Excel files, whatever, and get converted to text. And only Google Scholar and Cyrus, that's the Elsevier full text search engine, if you have never heard of it, have a collection of documents like that of a similar size. It's kind of funny that there's not a lot of effort of the research community to get a set of research documents. If Google ever decides to sunset its Scholar engine, then we don't have a search engine, a full text engine anymore for research articles. There's a website where I show more statistics on that and how many I get from which publishers, and I also have the list of people to contact for each publisher. That's a valuable resource. because I spent most of the time actually finding people within the publishing industry that are supportive of this project. You can have people that are against it and that are for you, and it's better to contact those that like the idea. So overall, I estimate that if you type in gene or protein into PubMed, I filtered it on the year 2010, then we have permissions for 80% of the results in PubMed. I've downloaded only a little bit more than half of it until now. And most of them are from big commercial publishers. So this pie chart here are only the big commercial publishers with more than 2%. The blue ones are the small ones. The rest ones, we have Springer now, and all the bigger ones except Wolters Kluwer. These are easy because these are just around 10 people you have to talk to. These are hard. These are the small ones, smaller than 2%. So more people to talk to, and if they don't reply, they don't reply. And then there are the really small ones, which we won't bother even contacting. So more than 85% is probably impossible, unless someone changes copyright law, which is about to change in the UK. So maybe if you know someone in the UK that might get very soon, that problem might simply disappear for people in the UK. There are a few technical problems that not many people know about. If you have 20 million files on a cluster, that doesn't go very well. So I packed them together into chunks. Processing in a cluster, there are people who use Hadoop. I can't really use Hadoop for various technical reasons, because genomics clusters are built and target genomics applications. So do astrophysics clusters. So each cluster has a slightly different scope. The clusters, the compute clusters you know from Facebook, Google, Yahoo, these all target text analytics. So they are perfect for that, but I don't have that. You cannot use databases. If you work on a cluster, you can't really use databases apart from Hadoop, very special databases. And many of my articles do not even have the most essential thing that you can use for analysis, the PubMed identifier, because all the databases refer to that. So I had to resolve that myself. And of course you have duplicates, and then when you get new data, you have to figure out what is the difference to the old data. So that all just takes time, and I had to write tools for that. And that might be useful for anyone who wants to do large-scale text processing afterwards. I have a few command line tools. They're command line tools, so they're not Java or Python specific or anything. A few command line tools on GitHub where you can type in, for example, pub get Elsevier, and then it will download everything from Elsevier that you have the rights to. Or pub get Springer, and it will contact a special Springer download server and convert them and process everything until you have, the output is always a directory of tables, Excel tables. You can open Excel or tab-separated tables. There's converters and downloaders for Medline, updaters, and also a crawler, which gets a PubMed ID and gives you the PDF. That took a while. That is used for the most of the smaller publishers. There is a tool, Quosa, that MGI is using, but I don't really know how much it costs, and I kind of didn't want to use a graphical user interface on the cluster. So these are all command line tools. And then there are others, the annotators annotate stuff, just entity recognition, DNA and protein sequences, and now I'm getting into more gene recognition and variants, mutations, various things. They run on the output. Again, command line tools, they get a directory and they spit out tables. Hopefully that's easy to use. And then in the end, everything gets loaded into the MySQL database and goes live on the UCSC genome browser. I also, so now you might be interested how much databases are covered at the moment. I cover most of the databases here, so when you create a paper, I have half of them, and I hope that will go up to 80%. It is increasing every day. So yeah, some of the older databases, the older the articles, still less coverage. That's the text collection. Now, what do I do with these documents? I want to map them to the genome, somehow link them to something that is on the genome. The easiest thing is accession numbers, something like NM underscore number, very weird identifiers that refer to other databases. So papers refer to sequences that someone else has submitted to a database. These are very easy to recognize most of the time, and there are hundreds of thousands in them. These are just counts, and you see that some databases are more used for that, more often cited than others. GenBank is the most common one. And I'm adding more and more of these databases. So these were just 7, and now there's so many databases that you can never stop. But I have regular expressions and expressions to find accession numbers for various other database, Flybase, Wormbase, MGI, SGD. If an author mentions the database, doesn't cite their paper, but mentions an identifier from them, I think we should have some flag or some alert system that tells us, okay, this database has been used. Now, a very special type of identifier is a DNA sequence, A, C, T, G, G, A, T, G, something, whatever string of nucleotides. It doesn't really relate directly to a database, but it indirectly refers to a gene. You can find it on the genome, and that's what I spent a lot of time on. I go over the text, find strings of nucleotides, and then search this on the genome and prepare this visualization on the genome browser where you have, that's the top part here on the track. So the chromosome is laid out left to right, always in the genome browser, and you have these matches in thick black and the thin parts that merge, join several matches that are flanking next to each other together, and mouse overs show the title. And then below you have the identifiers. They also link out to, they always link out to the sentence where the thing has mentioned. We are allowed to show sentences. We are not allowed to share the PDFs. We are allowed to have people run their software on the PDFs, but we are not allowed to give away the PDFs, of course. But we are allowed to show sentences. And thanks to the Yale image. Yes. So as long as it's somehow related to genome annotation, we can run anything on it and share the sentences. But we're not allowed to zip together all the 250 gigs and send them away. Yeah.
But it would be very useful for us.
Of course.
Over it and see what we missed or
Yep.
whatever.
Yeah. In figures, so one of the questions that often comes up, so what happens if someone mentions a gene on a figure? I don't know why that is so important, but it's probably important because if it's in a figure, the author cares about it. And I thought that's never possible, but the Yale Image Finder people have actually run optical carrier recognition on figure text from PubMed Central. And even if someone has mentions the gene or the gene sequence in a figure, so which is a bitmap, a graphic, I can still pick them up now, which is really nice, but only for PubMed Central.
That's really important, and I think the reason is because it's very hard to distinguish in a paper what, the discussion can bring up a lot of different terminologies than what's in the figures. I think the figures and materials and methods have been pretty well shown to be the core bit of information.
Okay. So the figures and the materials and methods.
Yeah,
okay. So, yeah. Mm-hmm.
Yeah, so that's right.
Okay, good to know. And then sometimes people have lists of things, lists of genes, lists of identifiers. Like if your database, you have the list of your identifiers, and you don't want to click 50,000 times and check all of the locations on the genome. So I hacked together this little batch interface where you can copy and paste a list of identifiers, and it returns how often that was cited, and you can find out how many just the sentences where that identifier has been mentioned. And then triage, so document triage. Apart from finding things in documents, I also want to assign them to topics of interest, sort them by subjects. And so the subjects, I think we already have because there are so many databases. Biological databases, each database is somehow a paradigm, a group of scientists, a topic by itself. MGIs, mouse, RGDs, red yeast, these are all groups of scientists. So we have these already. And each database contains somehow on its download section somewhere a list of PubMed IDs. Not always easy to find, but somewhere in the database there's usually a list of document identifiers. So I started to get a list of documents for a database like everyone else classification, and then ran this through a document classification, which I just copied from the Textpresso people. Wormbase is probably the most advanced when it comes to text mining. I just imitated their idea, which is get me the most relevant words for these document collection, for the documents, unigrams for text miners, and then use a support vector machine to classify. And I run this, I can run this now on 5 million articles, and it takes half an hour. So it's not, doesn't take too long. So this is just an example. The people that know something about bio-NLP classification, they're here not surprised that mice, of course, is the topmost word, but then you have all sorts of other weird words that come up. Some of them are the journal identifiers, because of course the classifier quickly learns that some journals are more important than others, and there are more words, there are 10,000 words. Sometimes I have ad hosting solutions. That is because I do HTML pages, I crawl, and then publishers sometimes link out. So it seems that mouse papers link out to have some special ads. And that's, of course, that's a bad example for a word that the classifier has learned that we don't really want in there. But there are thousands, so there is necessarily some noise. And it comes up with special years and knockout and, well, other words, names of lines. And the classification performance was surprisingly good. So this is upwards 90%. That might have to be with the fact that I use as a background not the MGI journals, but just the random articles. I just realized that with your discussion just right now, that you have a better or more realistic background set, and I should change that. I just didn't realize that when you gave your talk at biocuration. So I just use random articles, and there are yeast articles and whatever articles, and these are really easy to classify. So in general, from a genome browser perspective, that's good enough for us. We just want some topic. And I'm still adding databases. So to get these lists of PMIDs, I have human gene mutation databases, more immune genetics databases, and more human databases, of course, as a focus, more interaction databases I'm adding now. And for each of those, I have a list of articles that are likely related to the database, but not yet in the database. So I have tons of lists of PMIDs, and I'm happy to share these, or I want to share this. If you go to that website, then you have a list of all the PMIDs, bulk downloadable as zip file, and also the titles and abstracts. In the genome browser, the classifications are used for colors. So you remember the sequence matches used to be black. Now they're getting colored. Blue is something related to mouse or rat. Yellow is cis-regulatory, so promoter interaction. Green is biochemical, protein, that's mostly PDB. That's about it. And red is disease-related. So these are databases like OMIM with a long history of curation that somehow link a disease to some genomic locus. I'm doing other more involved things now. I'm getting into gene recognition. Gene recognition, I do that very differently from the existing one. So the existing gene recognizers use machine learning to find out if a symbol that they found somewhere, if the context around the symbol, mostly the words around it or the sentence, looks like one of the full-text corpuses, like the context in the corpus. I'm adding now a lot more context information, not only the sentence, but I'm trying to resolve it from a genome browser standpoint. We have all these databases, we have lots of information in there. I'm trying to throw all the information at the text. So that's an example, if the text mentions an identifier which is not clearly, according to entries, gene PKD1 is not clearly assigned to one single gene. It can be one of two genes because it has changed the meaning. And the authors mention in the text a primer, a little DNA sequence, then I know that PKD1, the one that was ambiguous, is not PRKD1, but PKD1. And if they mention, again, the gene bank accession, then I can say, okay, PKD1 is really essential. Well, if they mention the primer and the gene bank accession, it's a really important gene. What I want is not only gene recognition, but priorities for genes. I want to find the most important gene, the second most important gene, and I think it's a good idea to use this, to use all the additional information that we have to rank the genes. Ultimately, I want the topmost 3 genes to reduce the noise for my other steps. I have not really benchmarked this in terms of precision because I still have to import your corpus, but I know that on a corpus of 1,600 documents, I got around 98% recall. That doesn't really mean a lot, but on average, I restricted it to the topmost 3 genes. So it found around 4,000 to 5,000 genes in the 1,500 documents, and the topmost among those was always the one that the annotators choose to, almost always, apart from a few exceptions. So that's different than a normal gene finder setting benchmarking. Another thing that is important for the variant finder, and now we're getting really into genetics and genomics, and I don't want to spend too much time on it, but people sometimes specify things, sentences like the one on top here, R447H, and in biology there are tons of conventions like this. That's a mutation from R to H in a gene. And now I want to find out, is that possible? Is there actually really an R at position 447 in CLN2? So I can check that in software, and then I can map this position to the genome. So if you look for a mutation in a gene for a given human disease, the idea is that you can find these automatically. There are other programs that do that, kind of. Mutation Finder finds R447H, but it doesn't really check, and it doesn't map to the genome. So the genome mapping part here is the part that I'm doing. I need to do. And then there's, in general, with our big text collection and all these text mining things, and while I talk to databases, I figured that there's so many things that you can't do in Google Scholar or in PubMed because the query language is so restricted. You cannot look for sentences with 2 gene symbols and bind or binds. That would be, you need to find a text miner to get that done. And you cannot find for an MGI anatomy term. So there's many simple things that could be done, but are not done because text miners are the only ones that really know how to formulate those, and they need a text miner for it, but they're actually pretty simple. So I had this idea that if you just write 4 or 5 lines of codes and you paste them into a website, and the website runs it onto the PubMed Central full text and gives you the results, maybe that could help. I hope that is one way to get database curators to do a little bit of text mining. I still have to work the kinks out, but I hope that that's one idea. So I just ran a quick search for all words that end in omics. It's pretty simple, right? All words that end in omics. But you can't really do that with most of the standard search engines. I would like to, as the next steps, I would like to subclassify MGI in phenotype, allele. That's what you already mentioned. That got mentioned several times. For that, I have to understand the MGI download tables a little bit better. I need to find better benchmarks, and there's a few things like phosphorylation sites where biologists have come up with some sort of way to express it, like their own terminology that I would like to recognize better, similar to the variants. In principle, for each data type, you have to create a new type of text mining script each time. Sorry? Yeah. Okay, good. Good. Okay, and thanks to publishers and funders and Microsoft Research, who hopefully will give us many more documents very soon because they're running this on their internally now, if they can share the data. So they want to run this on the web. Thank you very much.
Just have a suggestion. So you might want to take a look at the
Kevin, hold on a sec. Kevin?
Just a suggestion, you might want to take a look at the GNAT system, GNAT from Arizona State University. So they do a lot of similar, so they're trying to accomplish a little bit different task of normalizing gene names to databases, but they make use of many of the kinds of things that you're doing, like looking to see is there really an R at position whatever, and they've got a bunch of other things that they tried that you might find useful.
Yeah, thanks.
Any other questions? So I have a quick thing that I'd like to ask. So one of the things that we discussed after the meeting, the biocuration meeting in Cambridge, was the opportunity to try and maybe disseminate models into database sites and such like. So and just seeing how well developed all of these things are, and actually your source code is on GitHub already for the command line tools. What are the next steps that you think would be to actually make that happen? So do we need to sit down and kind of like figure out the specifics of a project, or you know, what kind of interaction do we need to undergo to actually try to, because one of the ideas that we have is to try to actually see how we can leverage this resource so that we can then build classification systems that can be deployed. And so, you know, is it just a simple question that we have to sit down and design the models, build the models, then disseminate them? But
Seems easy to me. Because I'm not bound by any license restrictions on as the models. With respect to the models, there's no problem, right? So I can just put them on the internet and just add them to the GitHub account. They're small. They're pretty small. They're just Word and then some weight.
Right. So in other words, the thing that would be the determining factor would be to disseminate the execution engine. How does someone in a database actually use the trained model on an inflow of papers that come in? That would be the question.
How to run it, you mean?
Yeah, how to run it.
Yeah.
Okay. Great. Thank you.
So we just come up with an example command line or something like that.
Okay.
Yeah, we can do that.
Cool. Great. Thank you. So you're up. Just as an observation, there seems to be a lot of Jims and Jameses around in this meeting, so which is a good thing, I think.